So let's get that up there. And did that default? No, it did not. There we go. Okay, so last time we had left off on where we were talking about, notice that the cells with nuclei are actually a fairly recent development, almost 50% of the time of the development of Earth. And then the macroscopic organisms, anybody know what a macroscopic organism actually means? Does that necessarily mean like us? No, that can actually have quite a bit of meanings, right? Yeah. It just means that it has a macroscopic structure that they organize into, right? And require each other to survive within. And then we have like, dinosaurs, vertebrates, reptiles, birds. Dinosaurs and reptiles are very different things, by the way, in case you wonder. In some ways, birds can actually be closer to dinosaurs than reptiles are. Anyways, marsupials, primates, you get the idea. Here's human beings. Very, 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 very recently, right? So again, the multicellular organisms, these are eukaryotes. Some of the examples of these are animals, plants, human beings. Many macroscopic unicellular organisms, such as yeast, are also. Unicellular organisms are prokaryotes. And the big difference between these, probably the most prevalent difference, is that prokaryotes lack a nucleus, right? Okay, and you guys have all probably talked about this in more detail in other classes, okay? So we pretty heavily are going to draw from things like that. Please note that eukaryotes, being animals, plants, and such, and having unicellular eukaryotes, is kind of an interesting thing because there is a huge divide between a plant and most animals, right? And in terms of animals even, we share that same kingdom with things like an octopus, which is so far divided from us in terms of organization and evolution. That is amazing. Octopi have brains in each of their tentacles, right? And you can actually split those tentacles and cause them to grow into 2 new tentacles. So you're doubling brain mass? There's some crazy crap going on. Anyways, this is what's usually referred to as the tree of life right here. And so we can place these organisms into one of 3 major domains on what we refer to as the tree of life. And those are eukarya, bacteria, or archaea. These are going to be based primarily now on biochemical characteristics. But in the old days, we had to do them on other things, right? Because we didn't have really good testing methodologies, and it just took too long. But now, primarily, we use biochemical characteristics. DNA is one of the big ones, right? Okay. By the way, in case you're wondering what each of these things are talking about, you all probably know bacteria pretty well. That's usually not an issue. This is eukarya. We have corn here, which is the true ruler of the planet. The most advanced organism. Okay, so who do you think has more DNA? Humans or corn? Oh, definitely, by a lot. Yeah. Corn has 10 times more DNA than we do. Yeah, its DNA complexity is 10 times ours. In fact, that's pretty true for most plants, are way more in animals, because they can afford things like growths that are not productive growths, because they don't move around as much. We can't afford that. We had like a giant knot in us that was 1/10 of our mass. We'd be in trouble, wouldn't we? Yeah. Anyways, this is archaea right here. So one of the major molecules, macromolecules that we need to discuss, and we're going to spend a lot of the semester on actually, is going to be DNA. And this is deoxyribonucleic acid, right? This is constructed from 4 major building blocks. It's a linear polymer. It's composed of monomers that consist of those of 3 major groups, which is the sugar. In this case, because we're DNA, the sugar is going to be the deoxyribose, right? Notice the name actually tells you the whole thing, right? Obviously named by a chemist. If it would have been a biologist, it would have been somebody's name there, right? The Mikuzaki or Lewis molecule. So deoxyribose, what does the deoxy tell you, by the way? Locking an option, right? Is there something else that doesn't have a deoxy in it? RNA, yeah. Ribonucleic acid. And the only difference there is the oxygen, the OH group. And there's a RNA that does not transition into DNA. Yeah, there's a base, yeah. And we'll talk about that in a bit. By the way, there are actually more than 4 bases that are possible and can be created. But those are not actually standard life on Earth things. We won't get a whole lot into that because we just don't have time. I mean, we do not have time to cover everything. 5 credit hour course, and every single lecture I'll be like, we don't have time. Shit. Big subject. It also has a phosphate. You guys remember in Gen Chem, we're like, these are the polyatomic ions that you need to know. And there's phosphate, PO4. Guess what? This is why. Finally, the payoff. And then we have one of 4 nitrogenous bases. And these are just usually referred to as bases. To be honest, circular nitrogen-containing bases are the majority of bases in the organism. That's basically the base. This is adenine right here. This is, you guys probably are familiar with ACGT. Okay? So adenine, this is A. Yes, that's the A in ATP, ADP. This is, we conserve molecules in organisms, right? You don't want to have to make 20 different things if you can make one, okay? And so that's what happens in organisms. You'll see these recycled many, many times, which is why I'm going to tell you right now, flip cards, okay? You need to put these on flip cards, start flipping. Everybody knows what I mean when I say flip cards? Okay. You can use the app, though that doesn't work as well as flip cards. All right. Cytosine, and this is the C when we talk about DNA. Guanine, this is the G. Bless you. And this is thymine right here, and this is the T. Okay, does anybody notice any patterns happening here? Got it. Yep, C and T are single rings. Okay, how do these things match up? Does anybody know? A goes to T, C goes to G. So notice that every single time when we have these things matching up, we have 3 rings. And that's why they have essentially the same width. Okay? Not a huge amount of width. If we were to try to change one out to the other, it's very likely we would end up something different, right? Either 2 rings instead of 3, or 4 rings instead of 3, right? Okay. You'll see me do this a lot. It ends up burning some of our time, so I end up having to flip through things later. But this is that logic building, right? Okay. Where instead of memorizing, you understand. Okay? And that's why I want you to sort of follow along and try to reason through this before I get to it. And so the other thing you notice here is, how in the hell are these things held together? Anybody guess? Hydrogen bonds, right? And so what we're looking at here is that we have hydrogens bonded to an oxygen and nitrogen, right? In this case, nitrogens. Okay? And they're going to be the hydrogen bond donator. They're going to then bond to an acceptor. And what are the acceptor? They're lone pairs on an oxygen or nitrogen, right? And what you'll see is if you look very closely, these line up. Donator, acceptor. Acceptor, donator. See that? Donator. Where are we? Acceptors, right? Okay. Had to look. Okay. So this right here is the covalent part of the structure. This is the individual strands of the DNA. If you stretch these out, they're very long, but they're not normally stretched out. Know that these stretched out structures of polymer that we look at in the books all the time, total bullshit. Nothing to do with anything. They don't exist for any real length of DNA. They're here so you can see the damn thing, okay? This is not what it looks like. This is referred to as a backbone, okay? And does anybody know what types of bonds these are? You got it. Phosphodiester, right? I pointed that out a few times last semester, didn't I? I know, it's like I was sort of training you for this. Okay, so again, this is your phosphodiester bonds. Here's one phosphodiester. Here's the other phosphodiester, by the way. And then we have the sugars here. This is the sugar right here. And notice that they go back and forth, and this is our polymer. These are the monomer units, aren't they? Okay. Notice that this is kind of a weird polymer because normally we have the exact same structure for our monomers everywhere, right? In this case, we have what are usually referred to as side chains, okay? And these side chains can be different things, right? In many ways, you can think of this as a bit like a rope on a flag, and then you can attach different flags onto different parts of the rope, right? Does that make sense? Okay. So we have the rope, the polymer, monomers. These are just stuff attached to the side. Okay. And notice that the phosphate right here has a negative charge. Everybody see that okay? And so what sort of charge does DNA normally have? Does it? Do you think you have this really super negative things with thousands of negative charges floating around inside of your nucleus and like, yeah, so we have components of DNA that are not in here, right? We're just not looking at them, okay? For example, cations. Okay. And that's true for the proteins later, that's true for all sorts of things. We're looking at the organic structure of the DNA and we're ignoring the ions that would go with it, right? And that's because that's kind of variable and there's things that will happen there, okay? We'll touch on that, but we don't have time. I mean, there's so much in this field. Anyways, so is everybody okay with this so far? This is that kind of bullshit, but very useful mental model of DNA structure, okay? Okay, so how does DNA actually arrange in an organism that has DNA, of course? Do all organisms have DNA? Do they? Some have RNA only. There's actually kind of a loose definition of what's alive, living, and what's an organism. Like, there's a debate. Are viruses organisms? They can't reproduce by themselves, but with other organisms they can, right? And they're distinct life forms that evolve. There's an interesting thing that RNA predates DNA. Okay? And so originally there was RNA, then DNA developed as a storage methodology that makes RNA. And then RNA actually predates enzymes as well. There are RNA enzymes. Okay? And then they developed, a methodology was developed to take the RNA and go to proteins, right? We'll go into that process, but, okay. So this right here is your double helix structure. Again, this is kind of a BS way of showing it, but it's the unwound version of the structure that's the nicest looking for you to understand the helix, okay? And so what are you looking at here? You have two strands of your DNA, right? And they have bases. The bases are faced towards each other. Now, are they faced running in the same direction? No, they're faced in opposite directions, okay? So notice our sugars. These have a numbering schematic, right? Specifically 1, 2, 3, 4, 5. See that? And that numbering schematic is going to be used to determine which directionality we have on our DNA strands. Okay? That's how we're actually going to denote that. And so these two structures here, these are the molecular structure, this blue one. And then the red one's molecular structure is this polymer running along here, right? And then they're joined together by hydrogen bonding, right? Which is an intermolecular joining, isn't it? Now, what's the difference between that? Anybody remember your Gen Chem? How about the energy-wise? What do we call it? Huh? Are much weaker than covalent bonds, right? Like factors of 100 to 1,000, somewhere in there. Difference. So, what do we call it when we take water, break a bunch of its hydrogen bonds? So they're all joined together, 100% hydrogen bonded. We break 20% of those hydrogen bonds. What do we call that? It's not a reaction. So not hydrolysis, not dehydrogenation, none of that. We call that going from solid water to liquid water. What's that process called? Melting. Okay? And can you melt DNA? Yes, of course. Hydrogen bonds, you can melt them. Okay? Okay. So these are called Watson-Crick base pairs. Look up the history of Watson-Crick and what went on there. Not to diminish Watson and Crick. They did do work that was probably worthy of a Nobel Prize. But you probably should look up the history of that sometime. The parts you think they did not. They did not predict the structure. That was actually Linus Pauling, who, by the way, was like a good friend of my advisor and has a really funny quote that I think I've used on you guys before. I may not have mentioned it. It's him. You cannot understand a two-dimensional structure without building a model, a three-dimensional model. So, or you cannot understand a three-dimensional structure without building a three-dimensional model. Which is, I probably used on you. Buy your damn model kits. Okay. And then there was two female scientists that did the actual experimental work, and Watson and Crick stole that work. Yeah. Anyways, they did do their own work, though that was actually worthy of a Nobel Prize later. So, AT pairs from the hydrogen bonding. This is the AT pairing with your hydrogen bonding. Notice your hydrogen bond donator, hydrogen bond acceptor. Everybody okay with that? See, that has two pairings here, doesn't it? Okay. This right here is your GC pairing. It has 3 hydrogen bonds that occur. And notice again your matching here, right? What would happen if you tried to match this with one of these? I mean, it would hydrogen bond, right? No, it won't match perfectly, will it? So it's kind of like one of those keys that slides in, but doesn't hit all the pins, right? And that's what's going to happen if you ever mismatch. Will it hydrogen bond? Yeah. Will it hold it together as strongly? as if it had had the correct hydrogen bonding? No. No, it'll be energetically less favorable, right? And that means you're going to get a slight difference in the structure, aren't you? Okay? Okay, so the DNA structure, again, this is going to explain your hereditary and the storage of information throughout generations and within your own lifespan, right? Because these DNA templates are constantly going to be used throughout your lifespan. Does anybody know if people's expression of DNA changes throughout their lifespan? Okay, we'll talk about it in more detail later, but understand that there is the base coding of your DNA, okay? And that doesn't change that much between generations. The big thing that happens is you mix and match two parents in humans. Within your lifespan, your overall DNA shouldn't change that much. But the expression of it can change drastically depending on environment and what's happening, okay? And where you are in the body. And this is because we haven't talked about them, but there's all these things that are going to control which parts you can actually stretch out. Like I said, they're not just polymers stretched out, right? If that was true, then you have so damn much DNA, your damn single DNA would be like the size of your body. You'd have to have cells the size of a body. Yeah. Okay, so again, this is going to be the specific pairing of these bases right here, right? And what this does is that we can then use this patterning to create new strands of DNA, right? And this will theoretically at least create two identical daughter double helices from one parent strand. Okay. Now, the terminology here is actually a beautiful terminology that really works well. So this right here means that a parent strand always ceases to exist, doesn't it? And one half of the parent strand always ends up where? In one of the daughters, right? The other half of the parent strand ends up where? Yeah, the other daughter, right? And then each daughter is always composed of half new DNA, right? Okay. Now, interestingly enough, where do the mitochondria come from, everybody? The mother. The mitochondria are the mother's mitochondria. Isn't that kind of beautiful? Yeah. Come on, you gotta just enjoy the times that these things work, right? It's a beautiful representation. All right, so assume a single strand of DNA contains the 10 base pair segment, A, G, T, G, G, blah, blah, blah. You can get the idea. Read yourself. When the strand is paired with its anti-parallel strand, how many hydrogen bonds are present in this segment? And so what do we have to do here to figure this out? Exactly. You go back here, you go AT is 2. So every time A or T shows up, times 2, yeah, plus 2. And then GC plus 3 each, right? And then you'd go through 2, 3, 2, 3, 3, 3, 2, 2, 2, 3, right? Add them up. There you go. Anybody have any questions here? No? Okay. This is, they made this way overcomplicated in the clicker thing. But they do that all the time, don't they? Clarity's sake. All right, so again, you're going to have two complementary DNA strands. These will spontaneously assemble to form a double helix. Typically, what would happen is they're synthesized together, okay? But if you were to have two separated strands because you had melted your DNA, okay? They can theoretically join together spontaneously. though it can be a very fraught process that can take us a considerable amount of time due to the fact they're so damn long, right? Yeah. Anybody ever tried to zip a zipper and had it issues? Yeah. It's like that. Yeah. Okay, so the biochemical timescale equals the timescale for the biological interactions and processes. On the order of picoseconds to microseconds, you can get self-assembly, though keep in mind, like most processes like this is an equilibrium, okay? I'm gonna point this out now. Organisms are stacks of equilibrium that tip each other in different ways, okay? And that is biological processes. They're stacks of equilibria, okay? Or webs of equilibria if you want to make it look messier, which it kind of is. All right. So these actually can happen extremely fast, assuming you have all of the complementary cations and etc. Okay? Covalent bonds, here's the definition. Formed by electron sharing between two adjacent atoms, typically considered the strongest bonds. You can tell this is a biochemistry textbook. That is such a biochemistry way of looking at that. The typical CC covalent bonds have a distance of approximately 1.54 angstroms. This changes considerably if you have functional groups attached to them. It has a bond energy of approximately 355 kilojoules per mole. If you're talking about kcals, that will be 85. Okay. I'm gonna probably never say the kcal number again. Are you guys okay with just dealing with that? Okay. Okay. Remember that 1 angstrom is actually 1/10 of a nanometer. Okay. I'll be honest, I forget that quite often, embarrassingly often. So if I forget that, you guys can help me. Okay. All right. Which is 10 to the negative 10th meters, of course. Okay. A nanometer being 10 to the negative 9th, right? So an angstrom being 10 to the negative 10th. All right, so let's talk about resonance. Oh my goodness. The organic one is showing up again, and you guys are going to get this and more in organic 2. And oh my goodness, it's just like it never ends. That damn resonance. Okay, so remember, this is resonance right here. So we have many molecules that are going to show up, especially where we need them to be extremely stable and on-reactive. They're going to, like adenine, exhibit multiple covalent structures. These individual structures are actually called Kekel structures technically, but you can also call them resonance structures, which is no problem there at all. And so this all together, if you were to look at what this actually looks like, this is going to look like a mixture of these two structures, right? With essentially 1.5 bonds between each of these, right? And it shares that joined p orbitals, which we call pi orbitals, together over the entire ring structure, right? Okay. Anybody have any difficulty with that at all? Okay, there's actually a bunch of really interesting stuff about this. And keep in mind, this is also part of that resonance structure right here. That this has pi bonding overlap here, right? Okay, and so this is an entire conjugated structure, and this has aromaticity. Okay? Not that that terminology and concepts are going to get used to a huge amount of degrees here, but this does actually affect the reactions that can happen in your DNA. Okay? However, we're only going to very lightly touch on that. Okay, so ionic interactions. Ionic interactions are non-covalent interactions that will occur between fully charged atoms or molecules. The energy of electrostatic attraction between the opposite charges of repulsion between like charges would be given by this equation right here, which is the Coulomb energy right here. This is the radius, by the way, the distance between those two charged objects. Okay? Okay. K is the proportionality constant. Q and Q1 and 2 are the charges on the two atoms. The more charged they are, the stronger the charges on the atoms will be. D is the dielectric constant of your solvent. Keep in mind the different solvents will affect ionic attraction. Okay? One of the reasons, for example, water causes salts to dissolve, right? Okay. K is the proportionality constant, while R is the distance between your two charged particles. In this case, we can call them atoms. So let's talk about some electrostatic interactions in water. Typically, these electrostatic interactions will have a bond distance of approximately 3 angstroms. This gives you a pretty good idea that we're talking about a lot less energy and a lot looser of a connection, right? Keep in mind this is in water, though. A bond energy, this is going to be given by the Coulomb energy, which is 5.86 kilojoules per mole. Notice that difference versus our carbon-carbon connections. See that? Yeah, not quite a factor of 100, but pretty close, isn't it? Yeah. Okay. And again, keep in mind that these are the electrostatic interactions in water. Okay? This would be very different if this is salt, if a salt of it. All right, so these are the electric dipoles, molecules with no overall charge, as you guys probably remember. Specifically, electric dipoles are where these charges are uneven. So you have a semi-permanent charge that's going to be located on one end, which is the opposite of the charge on the other end, right? And that is a di-2-poles. Poles meaning positive and negative charge, right? And the dipoles can interact with other dipoles, of course, which are dipole-dipole interactions. Extreme versions of those we call hydrogen bonds, right? And they can be dipole-ion interactions, which is what we're seeing right here. For example, the water shells around most cations, right? You guys know what I'm talking about for water shells? Kind of? Okay. If you have an ion in water, what will happen is you'll have an arrangement of the water molecules which have a negative charge here and 2 positive charges coming out. And the waters will arrange around the ion, okay? Usually either 4 or 6. 6 is pretty common, but some are too small and it's only do 4. And what will happen is these then have all these positive charges facing out that get joined by more waters. Those waters can twist around and they form shells around the cations, okay? And they're semi-organized structures. And we're going to very much encounter this, especially when we talk about channels. What actually dictates channel function in a lot of these structures that we're going to look at is not the cation itself working with it. It's the cation plus its water shell, okay? Does anybody have any questions about this so far? Okay, this just shows some different arrangements. This is usually a stronger arrangement than this sort of arrangement right here, but you can also have this sort of arrangement, the stacking of dipoles. Okay. Hydrogen bonds, these are just very, very strong dipole-dipole interactions. They're going to occur between a highly electronegative atom and a hydrogen are covalently bonded together. This is then going to hydrogen bond to another electronegative atom. It's kind of a weird definition that your biochemistry book uses, but it kind of works. It's okay, I guess. Typically, you have a lone pair, right? And that's because that electronegative atom can concentrate charge in an area. And then you have a hydrogen, due to its short bond length, with that other electronegative atom. It has a very large charge on it, but it also is very small, so it can reach in very easily. Okay? And so you can get very, very tight bonds. Notice that bond distance for a hydrogen bond is actually really close, isn't it? 1.5 to 2.6 angstroms. 1.5 is like a carbon-carbon bond, isn't it? Which means they're really coming in together, and they can be quite strong. Okay? They're a bit easier to break than a carbon-carbon bond, but surprisingly strong for what they are. These typically have bond energies from 4 to 20 kilojoules per mole. You will note that 4 to 20, these can actually be stronger than ionic attractions. Okay? But they're considerably weaker than something like a carbon-carbon bond, a covalent bond, right? Anybody have any questions about that so far? No? Okay. Note that it does take some serious energy to go and break all hydrogen bonds in something, doesn't it? Okay? Okay, so let's talk about hydrogen bond donors and acceptors. The hydrogen bond donor is the group that includes both the atom to which the hydrogen bond atom is covalently bonded and the hydrogen atom itself. This would be your hydrogen bond donor right here. Okay? It's the one that donates the hydrogen in the hydrogen bond. The hydrogen bond acceptor is the lone pair of electrons. It's going to be the one that is less tightly linked to the hydrogen atom, and so that's going to be right here. You will notice that they look a lot like you're having a reaction, don't they? And theoretically, you can have a reaction. And that's what we call an acid and base. Right? That's just an Arrhenius acid-base reaction. Yeah. Typically, why you don't get reactions, these are too tightly bound. It's not a strong enough base. Or these are not strong enough acids. Strong enough acid here or strong enough base here. Okay? Okay. Anybody have any questions about hydrogen bonding? No? Fantastic. How about van der Waals interactions? Are van der Waals interactions rare? We don't talk about them a lot. No, they're just incredibly common, right? All over the place, all the time. Okay? So these are going to occur when two atoms are sufficiently close, or two molecules are sufficiently close. They'll occur when transient asymmetry and electron distribution in one atom induces complementary asymmetry in the neighboring atom. And so you get a positive charge on one end because the electrons happen to pop over on the other side. Okay? And the other atom ends up having the opposite happen, where the electrons pop over to the other side and end up with a little bit of a negative charge over there. Okay? And this happens all the time, right? Kind of like your hair. Your hair ends up a little on one side, a little bit on the other side sometimes. Happens, right? Just random probability. This is going to involve neighboring atoms attracting each other. They're very weak individually. Okay? They have bond energies anywhere from 2 to 4 kilojoules per mole, but these are typically when we're talking about pretty good sized ones. Okay? Individual molecule-wise, the lower end of the noble gas, xenon and on, can actually have some pretty significant van der Waals forces. Okay? But nothing like a hydrogen bond. When we're talking about a long chain like DNA, or we're talking about a 100 carbon chain, we're going to have some van der Waals forces, and they're going to be pretty damn big. Okay? When we talk about DNA, does anybody have any idea of the sort of sizes we're going to be talking about? We're talking thousands of bases, not individual atoms, bases. Okay? So we're talking some serious van der Waals forces here, but they're still going to be overshadowed because we have an ion on one side phosphates, and then we have a bunch of hydrogen bonds each one, right? So it actually has some really massive forces when we're talking about DNA for a molecule. All right. So the attraction is going to increase as two atoms come closer to each other until they're separated by the van der Waals distance. This is the van der Waals distance. It's the inflection point where you stop getting stronger attractions. Okay? At the distance shorter than the van der Waals contact distance, you actually start experiencing repulsive forces from the nuclei, and the electrons repelling each other. And those will increase exponentially. So that's why you see this really sharp uptick right here. Okay? Of course, as you get far enough apart, you get to the point where it just doesn't do anything. It's done. Does anybody have any questions? No? Okay, so let's talk about some properties of water. In terms of biology, water is almost synonymous with life, right? In fact, we don't even look for life where there isn't water, right? And so water is probably the single most important molecule in biology, period. Okay? Why does something happen? There's a pretty damn good chance that water is involved in that answer. Okay? Water is a polar molecule, meaning it has two poles. It's a dipole on it. There's a partial positive and partial negative end to the water. The hydrogens are the ones that are positive, though you can sum them up in a single dipole right here, right? Where the concentration of positive charge. And then the oxygen has the negative charge. Again, you can sum up that negative charge as being over here, but the truth of the matter is it has lone pairs that come off in a tetrahedral structure, right? So they're coming in and out at you. Okay? And those are actually where that negative charge actually exists. Okay? And that's those hydrogen bond acceptors. Water is highly cohesive because it has two hydrogen bonds for each water molecule can make, right? That adds up a lot, doesn't it? We saw that. That could be anywhere from like 5 to 20, right? 4 to 20-ish. Yeah, that adds up fast. A large number of hydrogen bonds are going to be formed in liquid water. The maximum number of hydrogen bonds will be formed in the crystalline ice. The crystalline ice can usually be considered approximately 100% hydrogen bound, but it's not actually. It's 90-something percent usually. If you had a perfect crystal, it would be 100%. Okay? And water is usually where you've broken 20% of those bonds. So 20% of the bonds give it some degree of motion, right? But it still has cohesiveness. And that's the liquid water. Okay? And then if you break all the bonds, that's the gas, right? Okay. Now, you don't actually have to break all the bonds to form the gas. You can actually still have a few hydrogen bonds here and there. Okay? Like 2 hydrogens can actually be floating around as a dimer. But that takes more energy to float. This is the hydrophobic effect. So when we are in an organism, when we're talking about an organism, everything is defined by its interactions with water, right? Because water is everywhere. Is there any part of you that doesn't have some sort of water interfering with it? Not a lot. Basically, 99.99% of the time, water is involved. Okay? What does that mean for this? Well, water has very strong intermolecular forces, doesn't it? And so if you don't have hydrogen bonding, what's going to happen? Everybody's played with Legos when they're a kid, right? They have like the magnetic pieces and the non-magnetic pieces. And you threw it into your little tub or whatever to clean up when your mom yelled at you or dad yelled at you. And so, the tub gets shaken around all over the place. What happens to all the magnetic pieces? They stick together, don't they? They end up clumped up. And basically all the non-magnetic ones end up getting excluded, don't they? Yeah. And so if the entire damn case is full of magnets, what happens? Yeah. Yeah, the non-magnetic ones are going to get squeezed all together, aren't they? And that's what happens in your body too, okay? This is the hydrophobic effect. Non-polar molecules in water are going to be driven together by this hydrophobic effect because essentially they're just going to get squeezed into one area, right? Excluded from the areas with water, okay? There will be an association boundary where you have the water molecules squeezing against them. And this is actually going to cause some serious energy, okay? Because it's not stable if you don't have something to interact with these water molecules. This is powered by an increase in entropy of water. Its associated interactions are called hydrophobic interactions. And these are incredibly important actually because yes, hydrogen bonding is part of these structures of your proteins and DNA and all that. But the other major part of this is actually hydrophobic interactions, which can be just as strong due to the excluding power of water, the fact they're squeezing them together, okay? And so much of the structure is actually going to be due to this hydrophobic effect. What types of atomic interactions do we see right here? Huh? Hydrogen bonding? Yep, hydrogen bonding, good. Anything else? Covalent bonding, right? Yeah. Okay, anything else? What's in everything? Huh? Yeah, van der Waals. Yeah. If it has atoms, van der Waals are there. 2 atoms, van der Waals. Okay? All the time. Even when they're bound together, theoretically you would have van der Waals forces, though I don't think we typically would recognize those as van der Waals forces. It's kind of a weird area where we kind of just go, hey, yeah, don't exist. How about ionic? Yeah, so theoretically, we have ionic repulsions here, don't we? Because we're not including the cations. If we assume the cations to be there, we also would have ionic interactions, right? And if we have water around this, which we definitely do, we have some ion dipole interactions, don't we? Yeah. So anyways, so the double helix is actually an expression of your rules of chemistry. You may not have realized it, but all of the rules that we've taught you up to this point have led to this structure, okay? And this is why Linus Pauling, he had the bases in the wrong directions. That's it. He came up with the whole structure of DNA theoretically from basic organic chemistry. That's it, okay? From organic chemistry 70-something years ago, more than 70 years ago. When a double helix forms, the charge repulsion is going to occur between the negatively charged phosphates of your backbone. So recognize that each of these phosphates are repelling each other, right? This can't accomplish much on the same strand, can it? Because it has the carbon and the covalent bonds linking them, okay? So it can change that structure only very little, okay? But when it has these overlaps, or we try to do a straight chain of this, what's going to happen? Well, they're going to repel to each other. So what structure ends up forming is the double helix when we try to reduce that repulsion, okay? And this is part of what forms that. These repulsive forces are going to be reduced by this high dielectric constants of our water and the interaction of positively charged ions with the phosphate groups. That's partially what you're not seeing here is there's waters and there's cations that are forming in between. And that's helping these repulsions minimize, okay? There's several different structures that can be formed, and there's also macromolecules that actually can help this structure become more stable, okay? But again, that's stuff we're not into yet. Why is if you just leave it alone by itself, double helix? That's why, okay? How's my time? Oh, okay. Okay. We'll just call it this slide here. We'll pick it up from next time. Okay, that's today. You all have a great day. I will see you tomorrow.